All right, well, good morning, everyone. First, a big thanks to both Prakash and Audrey for organizing and for allowing me to chair this first session. We have 3 talks this morning. I am told they're slotted for 30 minutes, so 25 minutes, and and then 5 minutes for questions. We'll get started with a close attention to time. We have 1 minute till we start. So our first talk is going to be from Manoj Duraisingh, who's a professor at the Harvard T.H. Chan School of Public Health, and he's going to tell us a bit about red blood cell genes.
All right. Great. Thanks, Jeff. Thanks so much to Prakash and to Audrey for inviting me to spend Malaria Day with this exciting focus on the blood stage. And so it's really great to be at Hopkins at the JHMRI. And so today I'm just going to talk to you about some of the approaches that we've been using. I put this title together a while ago, and I've been trying to think about what it means. And so this whole talk is going to be trying to interpret this title. And so as this is the first talk of the day, I think it is pertinent to just remember that malaria continues to be a huge disease burden, and control and elimination measures have somewhat stalled in the last 5 to 6 years. And I think many of us here really feel that new tools are going to be required to turbocharge the push towards elimination and eradication. Human malaria is caused by infection by apicomplexan parasites, protozoan parasites. Today, I'm going to talk about 3 of those: Plasmodium falciparum, Plasmodium vivax, and the zoonotic parasite Plasmodium knowlesi. They are evolutionarily distinct to their vertebrate hosts, which presents opportunities and challenges. Over many years, we've been using the life cycle as a blueprint for targeting the parasite in different ways. Targeting transmission is a hallmark of control. There's been great progress in terms of now actually targeting sporozoites in the liver stage of malaria. But today, all the talks are going to be focused on the stage that causes disease, this exponential engine that causes such a big parasite biomass within the body that leads to severe disease, which is why we're all focused on trying to eliminate it. So Benet, when she was in the lab, put together this nice diagram, which really just shows the intraerythrocytic developmental cycle. So a ring grows in a red cell, which then divides, forms lots of daughter progeny, and the cycle continues. We put this together because we wanted to quantify the different steps. And the idea was quite simple, that in the body, parasites per cycle grow a certain amount. And so the question was, if you were to actually limit it at different points in the cycle, how much do you need to limit it actually by? Is it 100%? Is it 80%? And so this was the notion there. And if you are able to limit that asexual proliferation, you might have a very decent shot at reducing the pathogenesis that I already told you was associated with very successful proliferation within the human. And this is an old study which is very intriguing because the y-axis here is actually invasion efficiency. And what it's showing is that parasites taken from people with severe malaria actually have a much higher efficiency of invasion of red cells than parasites taken from people with mild malaria. So this brings us to red cell invasion, which is the focus of today's talk. And so here is a parasite loosely attaching to a red cell. It apically reorientates, forms a tight junction, and then moves into the red cell. And actually, this stage takes the longest, it turns out, to actually form the vacuole within which it replicates. And there are lots of molecules, both from the parasite side and from the host side, that are responsible for mediating these different steps. And so if we could find those critical molecules, those essential molecules, is that me? That's what I sound like. Okay, so,
1 second.
okay, so, but the idea is if we can find those critical high affinity molecules, interactions, if we can target them, we might be able to prevent invasion and reduce that parasite multiplication rate. So I'm going to talk to you about 3 approaches that we've been using in the lab today to identify those essential interactions. One spearheaded by Ushir, a really great research scientist in the lab, who started looking at a major host polymorphism, which actually led to the identification of a novel ligand-receptor interaction. Mahmoud, who's another postdoc in the lab, has done an exciting functional screen that's revealed new host determinants, which we hope will lead to potential interventions. And then finally, we're going to actually talk about nullsci as a model system, which we're hoping will help us to understand how to target the vivax clade. And the overall hypothesis, of course, is if we can limit invasion, then and we can target those essential ligand receptor interactions at some point in the future. And I think today this is going to be talked about in the last session. We might be able to actually inform therapeutic interventions. So a lot of work has been carried out over the last several decades, which created this lock and key model where you have a lot of parasite ligands and keys on the red cell surface. And these tenets have been established that there's one parasite ligand for one receptor. These ligands are very polymorphic, they're variantly expressed, but then the variant usage of these ligands can actually lead to different tropisms for different kinds of host cells. But one salient point is that almost all of these, except for RH5, which I'm sure we're going to be coming back to during the day, can actually be experimentally deleted, which kind of makes you wonder whether those would actually be good targets for intervention. So, we're now in a post-genomic era. We've been in it for over 2 decades now. And, the parasite in the blood stage has some challenges. It's haploid, so working with essential genes to confirm that they're essential is difficult. The red cell is enucleated, another challenge. So, about a decade ago, we took up that challenge and started culturing and genetically manipulating red blood cells. And the idea was that if you could culture them, you start with nucleated cells that could be genetically manipulated to make mutants. And those mutants could then be used to analyze invasion and growth. And Dr. Amy Bay, when she was in the lab, used this approach very well to show that the glycophorin A receptor was actually used by some parasite strains, but not by others. So this is a knockdown of the glycophorin A receptor. And here you can see W2MF uses it, but 3D7 doesn't. And so then we worked with our friends and colleagues at the then called Sanger Center, Julian Rayner and Gavin Wright, who had used the biochemical means to identify the RH5 Basigin interaction. And so Amy, again, knocked down the Basigin receptor this time, using the same approach. And this time found that this interaction was used by all parasites for invasion. So it didn't matter what strain of parasite you actually had. So it was very exciting because we went from looking at these ligand receptor interactions as being strain specific to them being strain transcendent. So Elizabeth Egan, when she was in the lab, actually took a different approach, not a reverse genetics approach, but a forward genetics approach, and screened all the blood groups in a mutant pool and was able to find 2 genes that seemed to be required for Plasmodium falciparum to get in, CD55 and CD44. And that was important because they were also strain-transcendent, but they were previously completely unstudied. So we realized that there's actually a territory of discovery through using functional screens. And this was done in collaboration with the Broad Institute and Dr. John Dench. So we were super excited at that time. We thought, wow, okay, we have a system where we can now interrogate every interaction between the parasite and the red blood cell, and we're going to be busy. But, you know, this is what happens when you get too excited, and this was one of the reasons. And this graphic is informative and challenging, and this is what RNAi does. There's so many off-target effects when you actually use RNAi. But then, you know, the CRISPR came along at about exactly the right time and creates a specificity that was just not possible with RNAi. And so Ushir in the lab then used that and used Basigin, that receptor that was shown to be strain-transcendent, as a case study and knocked it out. And here you can see that's a clean knockout. Nothing else is the surface proteomics. Nothing else on the surface changes except Basigin. And now parasites just don't get in at all. And this is true for all the strains that we've actually tried. This is the knockdown just for comparison, because it was only a partial knockdown. There was still some invasion into those red cells. So Basigin is essential for strain-transcendent invasion, and then in really exciting work, which I'm sure we're going to hear about later, you can actually see that the RH5 Basigin interaction is being pursued. So we now have a toolkit in the lab which uses primary cells for genetic manipulation, but we also use these immortalized cells, which allow us to essentially do very stable genetic manipulations in scale. And so we've assembled all of these methods in the lab, and that helps us to interrogate the red blood cell. So Ushir decided to look at the question in this way. Can we look at natural polymorphism as a means to actually identify important determinants for malaria infection? And this was known for a long time, that sickle cell trait has been, has risen to high levels in malaria-exposed regions. But there's also significant variation on the surface of the red cell. It's sometimes thought to be the most polymorphic surface of all cells in the body. And when you look at this, they're actually in 3 blood group receptors. Here are just 3 examples. The Duffy receptor, which, Dr. Lou Miller has done amazing work showing is really required for Plasmodium vivax to invade. And then more recently, glycophorin B, which the DAN2 polymorphism seems to afford a protection against severe malaria almost as strong as that of sickle cell trait. But Ushir decided to look at BAN3. So BAN3 SLC4A1 is polymorphic in malaria endemic areas. It turns out it's the most abundant surface on the red cell surface. And there's this polymorphism, which is a small deletion which leads to Southeast Asian ovalocytosis, which has risen to really high levels in some regions, sometimes up to 30% frequency. What's amazing about this polymorphism is you only see it as a heterozygote. It's actually a homozygote lethal. I think there have been 6 people who have actually been born with homozygous at 27 base pair deletion. So the question is, why was this selected for? The SAO cells, it turns out, these are heterozygote cells, are less deformable, and there's reduced falciparum invasion. And it's been associated with clinical protection from falciparum malaria. And those are the ovalocytes. They are actually oval. And so Ushir used the same in vitro culture genetic system to delete BAN3 from these erythroid lines. And so you can see here, BAN3 is completely missing. Didn't even know whether this would be possible. The cells actually are a little bit smaller. This is volume, so they're only a little bit smaller. You can see the cells here. And then we looked at tension because Dr. Julian Rayner had found that tension is a pretty strong determinant of invasion. And these erythroid cells, red cells, are actually, they have a lower tension in general. So actually, this is a higher tension in general than peripheral red blood cells. And when you knock out BAN3, it actually increases. And so there is actually a significant difference here. So it is possible that this is a determinant of invasion. But Ushir did another experiment, which is he then took lots of different parasite strains. And what he did is he just looked at invasion efficiency into these cells. And he found that all strains that he tested were inhibited in their ability to invade. And there were some differences, slight differences, like W2MEF was able to invade a little bit better than these lines, and these two field isolates a little bit less well. So the question was, well, okay, it looks like all strains use it, but still, because there are slight differences, it might be that there's a ligand-receptor interaction involved. And so this was a very exciting experiment that he did, and it's quite difficult to follow. But he took a lot of parasite ligand knockouts, and he essentially used them on these wild-type and BAN3 knockout cells. And what we found was that inhibition was observed in every single knockout except the EBA181 knockout. So there's a genetic interaction between BAN3 and falciparum, the EBA181 invasion ligand. Now the question is why? And this is a model, which is what we think is actually happening, that if you have either the ligand or the receptor missing, you actually have reduced invasion. But if you have both the ligand and the receptor missing, you don't get any further reduction in invasion because all the other ligand receptors interactions actually take over. So what do you do next is you actually do biochemistry. And so he essentially did an EBA assay, which is taking parasite supernatant and incubating it with red cells. In this case, we took the mutant erythroid cells, the wild type and the knockout cells. And what he found, what you do is you basically spin those cells through some oil and you elute them, and then you probe to see what had actually bound to those red cells. And then what Ushir found, which is very exciting, was that that EBA181 binds to the wild type cells but does not bind to the BAN3 knockout cells, suggesting that BAN3 is actually a direct receptor for EBA181. And then when you do surface proteomics again, you find that no other receptors actually change significantly on the surface of those mutant BAN3 knockout red blood cells. And so we believe that we might have an additional ligand receptor interaction, which is used by all strains that we've actually tested so far. So Mahmoud in the lab decided to do a functional genetics screen, which is a parallel to that RNAi screen that I showed you, but now through using the power of CRISPR. And so again, it's a pool screen where he created a pool of mutants, this time all the membrane proteins, because we had already done proteomics and it found about 200 proteins on the surface of red blood cells that we had high confidence in. And so here are the mutant pools. You compare essentially invasion into the mutant pools versus the mutant pool itself to see, and then you sequence each of these pools because each mutant is barcoded. And if there's a difference, that actually should reveal the cells that were unable to be invaded because they'll be missing from this pool. So he did this, and we were really excited to see that some hits really stood out. And so, first of all, Basigin, thankfully, and CD55, two things that we had found previously, were really high hits in this screen. And then I validated it, and then we also found these two genes were also in the screen, and these had actually popped up as top hits in an RNAi screen previously, and Estella in the lab had actually done more work on it. But then you notice that there are actually two new genes here, which we had not actually previously seen, except for the fact that one of these, interestingly enough, is in a complex with a protein, which is actually a receptor for a P.vivax ligand, PVRBP2A. So this gene came up in our screen, so that was intriguing. And another one, which really looks like a receptor, it has a nice big ectodomain here, TMEM30A also came up. And that one's kind of interesting. You know, it's thought to be in a complex with Basigin. It's come up in the SARS-CoV-2 world. And there's even some evidence that it has a role in liver stage invasion by falciparum. So Mahmoud then validated these using the primary in vitro culture system by knocking out each gene individually in primary cultured red blood cells, and saw that the invasion defects were replicated. And so we now think we have two new host determinants. Are they receptors? We do not know yet. We'd have to find their cognate parasite ligands. Are they working in another way? Is this revealing alternative mechanisms in which invasion can be inhibited? And that's where we are with this story. Overall, what we're really finding is that there are a bunch of ligands that are strain-specific, receptors that are strain-specific, another group that are strain-transcendent. We think the strain-specific ones are very important in pathogenesis and maybe even in transmission, but the strain-transcendent ones, of course, because they're essential for invasion of all parasite strains, we think would be useful in terms of following up for in vaccine development. And so we've also now identified new RBC determinants, and here's the invasion team who are following up the genetics by now trying to understand the mechanistic basis by which each of those genes are actually affecting their inhibition. So just to finish, I'm just going to talk to you about nullside very quickly. So Ushir has spent a lot of time working on Vivax, which is not for the faint-hearted at all. And so beyond the PV-DBP-Duffy interaction, they actually found that PVRBP2B binds to the transferrin receptor. And so that was exciting. And Ushir's contribution was that he was able to create a knockdown of the transferrin receptor, and he showed that Vivax cannot actually invade those cells very well. But this is really tough work because there's no in vitro culture system yet for Plasmodium vivax. So Sheena actually decided to take a different approach here and to actually think about whether or not the members of the Vivax clade, so here's Vivax, cynomolgi can be cultured, and nullside can also be cultured, whether their shared, their evolutionary closeness would actually mimic, would actually result in a shared antigenicity on their surface. And so in a great collaboration with Professor Julian Rayner at CIMR and Dr. Marcelo Ferreira at USP, essentially Prasoon and Deboki have created antibodies against a bunch of Vivax antigens. So these are against Vivax antigens, and they all have different levels of homology with cynomolgi and nullside. And so the idea was to see whether these would also inhibit nullside and cynomolgi. These are Vivax-focused inhibitory antibodies. And so we miniaturized these assays. And yes, they do. They actually inhibit cynomolgi and nullside quite well in similar ways. And falciparum was a very nice control because when you use the same antibodies against falciparum, there's almost no invasion at all. And now, in a big project, Prasoon and Deboki in Julian's lab have actually made antibodies against 120 predicted Vivax antigens. And we're using this approach to actually try and screen them. And to validate this, we actually took one of them. So there's antibodies against this new antigen PVP108. And we did do that painful Vivax inhibition experiment. And we actually did find that the one, that antibody actually inhibits at a similar level Plasmodium vivax as it does nullside and cynomolgi. And so this is a screening strategy. We're not saying, it's a practical screening strategy which we think might be useful in identifying candidates for future Vivax inhibition assays, particularly if you're starting with large amounts of sera. So just to finish off with, I'm just going to give you a very quick run through to another model that we've created in the lab, because again, we think null's eye, because of its superior genetics, and this is what Adi has found, that if you zap them, almost 10% of the parasites actually are quite happily holding on to their plasmids, and that's what this is. And so based on that high efficiency, we decided to try the piggyback transposon, which John Adams' group has done wonderful work with in Plasmodium falciparum. And we did a simple scheme where we just took the transposon and the transposase plasmids, zapped them in, and this is what we found. And this is work started by Brendan, who's now in your area at the FDA, and continued by Sheena, but a great collaboration with these computational biologists at UMass Boston, Sita Ye and Dr. Karusha Ringalam, who's a great friend and colleague. And we had amazing insertion frequencies, and that's why this looks like this, 160,000 independent insertions, straight away gene level saturation. That's what this indicates. This is just showing, it's a model showing how quickly you get saturation into non-essential genes. What do you find? You find something very similar to falciparum. There's a large number of essential genes. This is an MIS score, so that's the mutability index score. And so essentially, if it's very high, it's non-essential. If it's very low, it's essential. So there are a lot of essential genes in null's eye. And this is when you actually look at the MIS scores from the falciparum screen that was previously done in the Adams group and our null's eye screen. We find that these peaks, this is the mutability score index, are actually shifted to the left. This suggests that because of the high degree of saturation, we have much greater confidence in being able to say that these are essential. And that's really important because it allows us now to use these pools in perturbation studies. We can map any gene onto this curve. Again, these are mutable, these are not mutable, essential, non-essential. When you do it with falciparum, you find that at the essential end, it doesn't look so, they look like there are a lot of differences. And when you actually look at the ones that are predicted to be essential and non-essential in falciparum and null's eye, you see a lot of discrepancies. So the question is why? We have no idea. It could be divergent biology or it could be different discovery rates. And finally, just to tie it all back to where we started, we can use this now to catalog invasion-related genes. Many of our favorite genes that encode antigens that many of us are interested in are actually now found to be totally essential. And this allows us to quickly prioritize. So just to finish, we're able to identify essential genes both in parasites and in hosts that define ligand-receptor interactions that can be strain-transcendent, helps us to prioritize those. We've found new factors. If we can find the ligands, they could potentially be good vaccine candidates. And we think null's eye and cytomology are great model systems that should be developed for different reasons, ranging from being surrogate models for invasion inhibition, as well as powerful genetic models that allow for perturbation studies. So that's the mantra of the lab. We're hoping we can get these essential ligand receptor interactions, and most importantly, here is the lab. This is all their work, and our collaborators and our funders and all of you, thanks for listening. Sick.
Thank you. That was a wonderful talk. I was curious about something with your band 3 story. You mentioned that within the heterozygote knockdown, that the tension of the membrane dropped. And there's a lot of interest, as I'm sure you're aware, of mechanosensing ion channels in promoting and controlling invasion of various cells. So my question is when the parasite enters a red cell, does it change the tension of the membrane? And is the problem with the low tension mutants that the parasite simply can't increase the tension high enough to open the mechanosensing channel and thus cannot invade the cell?
Okay, so that's an interesting question. Thanks. So the idea is that once a parasite gets in, does it transform the red cell and then prevent reinvasion because it's altered the tension of the surface? The answer is I don't know. I think it's an interesting idea. Certainly the red cell gets transformed. I think Dr. Deitch is going to be talking about that very soon. And I think determinants of reinfection or superinfection are really interesting. We haven't really explored that directly yet, but I think we could devise a screen to address that if we looked at invasion into already infected red cells.
I wanted to make a comment about one of the lines you're using. It has been very important that Moon from England has passed this out to everyone with no strings attached. That's what science should do. And I just make that point because he's doing what everyone should do, is if you have a line that's so important, he has a Knowles-eye line with a P.vivax receptor in it, and he's passing it out to everyone. And when you write him, he doesn't say, oh, you have to put my name on it or this. He just says nothing. He just have a good time.
Yeah. I think Rob's a great guy. I will have to say one thing though. This was actually done in macaque cells using a different H line, one that we actually culture adapted ourselves. And the reason being that the efficiency of transfection and growth is actually higher when you grow Plasmodium, even human-adapted Plasmodium knowles-eye in macaque cells. So we actually always use macaque cells because we can in the US of A. So